We ready to bring the jury back in? Mr. West, you ready, sir?
Yes, Your Honor.
Okay, thank you. Let's go ahead and bring him in.
Jury's present, Your Honor.
Please be seated. Mr. West, you may proceed.
Thank you.
Good afternoon.
Good afternoon.
Do you have this chart in front of you?
I do.
Okay, so if we refer to that, you've got a reference.
Yes, I have my reports as well.
When you say reports, you're talking about the summary findings?
The 3 reports I issued with all the results
Right.
that are in this chart.
Okay, and do you have your notes with you if need be?
Yes.
Great, okay.
203, Your Honor. Which one is it? That's 203.
The chart, thank you. The chart is exhibit number 203. Is that what yours says?
Yes, it has a 203 on it.
Okay. What I'd like to try to do is sort of track through the evidence a lot in the same order that was just done, so sort of keep that sequence and that frame of reference.
Okay.
But before, let's talk a little bit, Joe, generally about the DNA work that you do. It sounds like a lot of the analysis that you did was based upon evidence collection that someone else did. For example, swabs from other items.
That's correct.
So you would have played no part in collecting whatever evidence may exist from that flashlight, for example.
Correct. I don't actually go to scenes and collect evidence. I've actually never been to a scene and collected evidence. I first see the evidence when it comes in to the laboratory and brought to me in a sealed envelope or a sealed paper bag, and I take it from there. Sometimes it's a swab that someone else collects. Sometimes, as in the jacket and sweatshirt, sometimes it's the actual item.
But it's always gone through someone else's hands first.
Yes, that's correct.
And in this instance, there were swabs of the firearm of 4 different sections of the firearm.
Yes.
So you don't make any determination, then I take it, where DNA should be and isn't, for example. You just report what you find.
Correct. In that instance, I just test those 4 swabs and report my results from those 4 swabs.
For example, it's not in dispute that George Zimmerman fired the gun, but there was no DNA on the trigger. You don't try to figure out why that might be.
That's correct. I just report out that I was not able to obtain any DNA results from that swab from the trigger.
Sometimes you can touch an object and there not be any DNA left behind.
That's correct.
Sometimes if there might even be blood, it could be wiped off.
Yes, that's correct as well. Items can be washed and cleaned, wiped, washed, and that could affect my ability to get any DNA from touch evidence or blood evidence on an item.
That might include environmental factors, whether or not it's raining, for example.
Yes, moisture, heat, humidity, all those environmental factors can degrade DNA, meaning it chops the DNA up into pieces. The DNA is a long chain with letters and those letters, segments of those chains, those are those 13 loci that I test. So if those segments are degraded, those short segments are degraded and in pieces, that affects my ability to amplify and make copies of those short segments and be able to see any results that I can make any kind of comparisons with.
You're suggesting that humidity, specifically rain, can do that?
Moisture, which involves humidity and rain.
And of course, it's possible, I suppose, on a gun, for example, that because it's a hard surface, it could just be wiped off because someone handled it.
That's correct, yes. A gun is a hard surface, so any touch DNA or skin cells is going to be on that hard surface. There's no medium for any skin cells to soak into it. As, for example, on a shirt, sometimes if I'm trying to determine who's wearing a shirt and I'm trying to get skin cells from like the inside of the collar or the armpits that come in contact with that individual, that shirt could soak in those skin cells. On a hard surface like a gun or a knife or a pen, I'm trying to get what's on that surface. It's a hard surface and it's not good for collecting and retaining touch DNA.
So, for example, on 21C, the pistol slide that we talked about a minute ago, your conclusion was that there were limited DNA results and you were not able to make a determination regarding Mr. Zimmerman or Mr. Martin.
Yes, 21C from the slide. That's correct.
Which means that there was some DNA that you could identify through analysis.
Correct. There was some information present from my testing of that item. It was not enough information for me to be able to match it to any one individual. I can't tell if it's a mixture or single source because a lot of that information that I need is missing. But there was enough information for that specific sample for me to make comparisons and make an exclusion, potentially. And I wasn't able to exclude either of the two individuals.
So what that means, bottom line, is that in some situations you have limited DNA, but you have enough to say it could not have come from someone.
Correct.
Whereas in this case you had a limited amount, but indeed the DNA could have come from either George Zimmerman or Trayvon Martin.
I wasn't able to exclude them, so I can't say that there's no way that they could have contributed to that very limited sample.
Mm-hmm. Of course, the slide of the gun is also a hard surface that would be subject to not only the same environmental effects that we talked about, but perhaps handling, the way the evidence was packaged or secured even.
Correct.
Similarly, with regard to the holster, you did indicate that you were able to identify a profile that matched Mr. Zimmerman.
Correct. There was
I'm sorry. So you're confident then that Mr. Zimmerman's DNA is on the holster?
The major DNA profile from the holster matched George Zimmerman.
And you wouldn't know from what part of the holster, for example?
No, I don't know where on the holster they swabbed. It was represented as coming from the holster, so I don't know if that means the entire holster, the inside, outside, or what. Just
somewhere on that greater surface, there must have been some of George Zimmerman's DNA somewhere.
Yes, the major profile matched George Zimmerman.
And then insofar as the holster from Trayvon Martin, you had limited information on a minor contributor?
Yes.
And you were not able to exclude or specifically include Trayvon Martin as a contributor to that DNA?
That's correct.
When you say exclude, you mean could not possibly have been in your world. In the scientific world of DNA statistics, when you exclude somebody, you're saying, with those kinds of numbers, it really couldn't have been that person.
No, I'm saying there's enough of an absence of their information that they could not have been part of that data. That data that I'm seeing, that mixture of DNA, if I exclude someone, I'm saying they could not, I don't think they contributed to that data. Does that mean they never touched or came in contact or seen that item? I don't know. I know from what I tested on that swab and my results, if I exclude someone, I'm saying I don't think that they are possibly part of that data that I obtained from that swab.
And when you say you've excluded them, it's based upon that that probability statistical model that you talked about?
Not necessarily.
No?
I'm saying I don't feel that their information is not represented enough that I feel there's no way that they could have contributed to that data. I perform the statistics for the inclusions and matches to show relevance for a match or an inclusion, but an exclusion is just, I don't feel that that person could have contributed, and that's the end of it.
Not enough data. Well, of the data that you have, it simply doesn't match in any meaningful way the person that you've excluded.
Exactly. The data present does not enough of that person's profile is present in that unknown sample for me to say they could have possibly contributed to it.
Does the ability to conduct reliable, meaningful DNA analysis sometimes also depend on the way the evidence is collected and packaged?
Yes.
So, for example, if the collection was just not a good job, there could be DNA that was missed, and you wouldn't know that.
Potentially, if an item is collected and not stored properly or packaged properly, it could be exposed to those environmental factors that I discussed earlier, like heat, humidity, bacteria, microorganisms. All of those can degrade DNA on a sample, on a gun or a knife or whatever sample we're talking about. And once that damage is done, once that DNA is degraded and in pieces, that's going to affect my ability to see results when I look at that DNA profile.
It's very difficult for you to be able to figure out how much damage was done. It's hard to know what you don't know, in other words.
Correct. The first I see of the evidence is when it's brought to me in the laboratory, the package. I don't know when that was collected, how it was stored, how long it was sitting in an office or wherever before it was submitted to our laboratory.
I think the next item that you talked about were swabs that were collected. I'm sorry, these little Exhibit 169 are the swabs from the flashlight that had no DNA results, correct? And also the swab from the bag of Skittles candy?
Correct.
And similarly, you received the swabs, someone else swabbed the item itself and gave this to you, and you did your analysis.
Yes, I never saw the bag, the Skittles bag or the flashlight.
Now we have Exhibit 191. Those are the little wooden sticks that you talked about that in your training and experience are used for fingernail scrapings.
Yes.
And what your understanding of the procedure is that the small stick is scraped under the edge of the fingernail to see if whatever's under the fingernail that might have some biological component is gathered on the stick and then submitted to you.
That's correct.
So I take it could be blood, but it could be other stuff too?
Well, if I see red-brown staining on the item, I do test it for the possible presence of blood, which I did with the right hand, the stick collected from the right hand, and that gave me a positive result.
Yes.
So
I'm sorry, I didn't mean to cut you off. You actually saw something on the stick that made you think it could be blood?
Yes.
And then you did the presumptive?
Yes.
And then you got a positive, so you did the DNA?
Yes.
And you indeed were able to confirm that that was Trayvon Martin's blood?
Yes, the profile matched him, which means nothing foreign. I did not find anything foreign to him on that stick.
And then on the left, I don't want to confuse them, the Which one was negative?
The left hand, I did not test for blood. There was no staining on the stick collected from the left hand, and I was not able to obtain any DNA results from my testing on that stick.
So even though you didn't see what in your mind might be blood, you tested it anyway?
Yeah, well, I'm not necessarily looking for blood. A lot of times when fingernails or fingernail scrapings are collected, there's going to be blood present. A lot of times that person is bleeding, touching that area that they're bleeding from, and their own blood a lot of times is going to be present. I'm more interested in anything foreign to the person whose fingernails they are. So I'm more interested in any maybe skin from the other person or other people involved in the incident that might have scraped off underneath the fingernails of that individual. It could be blood from that other individual. If there was a fight and that other person was bleeding, there could be blood from both people underneath the fingernails. But I'm more interested in anything foreign to the blood from that person.
Insofar as the blood on the right hand from Trayvon Martin, you would have no idea when that blood may have come in contact with his fingernail.
That's correct.
Whether it was during whatever incident took place or after, if he touched his own clothing that had blood.
That's correct.
Or transferred from during the process of delivery service, for example. In other words, you just don't know.
That's correct. I just know whatever was on that stick when I swabbed it, that's when those are the results. I don't know when whatever DNA got underneath those fingernails. I don't know when that happened.
And on the left hand, there was no DNA of anyone, not even Trayvon Martin.
That's correct.
Did that surprise you?
No.
Because there just wasn't? Is that what it boils down to?
I mean, it's a very small stick, and you're scraping underneath fingernails. I can expect to see the DNA from that person whose fingers they are. However, it's not a guarantee that I'm going to get DNA from them. Most of the time, you see at least some kind of DNA profile from that individual, but it doesn't surprise me if I get no DNA results. It's a very small stick that I'm swabbing in a very small area that they're trying to collect DNA from.
That's something that you've seen before, where there just hasn't been any DNA on the fingernail scraping stick.
That's correct.
So in other words, the absence of evidence isn't particularly significant to you in that instance?
No.
Have you, I take it that you were not present when that process was done.
I was not.
So you didn't see anyone do any scraping, and I take it you never saw Trayvon Martin's fingernails.
No.
You don't know if they were long or short or whether there was anything at all visible.
No, I have no idea.
There is something curious here, though, that I wanted to ask you about. There's only one each, one for left and one for right. Do you think in evidence collection that all 5 fingers on each hand would have been scraped with the same stick?
Yes, generally speaking, that one stick would be used to scrape all 5 fingernails from that hand.
So you would take the stick and you would pull it underneath one fingernail, and then you would take the same thing and pull it under the other, and then likewise for each hand?
I've never done that process. I've never worked in the capacity where I would be collecting that type of evidence, but I would imagine that that's how they would be doing it. But again, I would just be guessing because I've never done that.
Oh, I'm sorry. So you sort of just, in your mind, common sense would tell you that one stick would be used for all 5?
Yes.
As opposed to maybe only one fingernail was scraped on each hand?
I mean, that's certainly a possibility. However, they represented that from being the left hand. So in my mind, they scraped all 5 fingernails from that left hand and from that right hand with those 2 sticks.
If this matters to you, on the packaging it says fingernail left and fingernail right, as opposed to fingernails. Does that mean anything to you?
No.
You didn't participate at all in the collection of this, as you said. Well, we can talk to the people that did.
Yes, that's how they chose to label. And type it out and put that label on there.
I noticed, though, just curiously, that each part of the gun was swabbed separately. Do you remember that?
Yes.
So there's a swab that relates to the slide, a separate one for the grip, a separate one for the trigger, and then a separate one for the holster.
Yes.
So that would tell you where, at least from what part, more precisely, any DNA was located.
Yes.
So, for example, on that that right hand, I guess, where Trayvon Martin's blood was, you would have no idea under which nail or where that was actually collected.
That's correct. Somewhere on the right hand, that blood was present, at least under one of those fingernails.
According to the packaging.
Yes.
All right, let's talk about the hooded sweatshirt for a moment. I believe that's Exhibit 155. That's represented in Exhibit 118 in the picture. Let's talk about the packaging of this particular item, or in fact, well, first, did you do any processing for DNA of any sort on Trayvon Martin's pants?
No.
Never saw those.
No.
So your DNA analysis was based upon two articles of clothing, the hooded sweatshirt and then the underneath shirt?
As far as what Trayvon Martin was wearing?
Yes.
Yes.
Mm-hmm. So as far as those particular items, you talked a little bit about packaging. Packaging that can, by virtue of how it's packaged, cause degradation of biological samples.
It could potentially, yes.
You're familiar with NIST? Do you know the acronym NIST?
National Institute of Standards and Technology.
Right. That's an organization that helps standardize such things as evidence collection techniques and such.
Yes. NIST is a government organization that issues standards. We actually have the standards for like calibration and stuff like that as a reference to make sure that your instrumentation works. We actually have NIST standards in our laboratory that we're required to run annually, and sometimes more than that, depending on if we get a new instrument or any new chemistry, to make sure that we're obtaining the right results from these standards.
And is NIST part of the accreditation process for the lab?
No.
Then you mentioned that the lab is accredited.
Yes.
But that's a different set of standards than the NIST standards that you were talking about?
That's correct.
Explain that just a bit.
We are accredited by ASCLD Lab. That stands for the American Society of Crime Lab Directors Laboratory Accreditation Board, and it's an organization made up of forensic laboratory directors from all over the country, and they come out and they do a thorough inspection of your facilities, your standard operating procedures, your case files. They interview your personnel, and they make sure that you meet the FBI's quality assurance standards in order to maintain or get accreditation. That inspection takes place every 5 years. We had one in 2010, just 3 years ago now. And in between those 5-year inspections, we're required to maintain accreditation. We're required to undergo an internal audit every year and an external audit every other year.
And in addition to that, for the lab, you also yourself have to undergo regular proficiency testing?
Yes, that's part of the accreditation process that every analyst or forensic technologist that wants to be proficient in DNA testing needs to undergo those proficiency tests that I discussed at the very beginning of my testimony. Tests from outside companies that I run. It's a set of samples that I run as if it were a case, and I send the results out, and then they send a notice back saying I passed or I didn't pass, and that I do every 6 months. Everyone in the lab is required to do that every 6 months.
Sounds like at least then 10 times each 5-year cycle that you yourself have to go through proficiency testing.
Yes.
When we're talking about standards and we're talking about biological evidence in particular, is biological evidence sort of a broad term for what may contain blood, fluids, DNA, and that sort of thing?
Yes, it is. I work in the biology section, so we don't call it the DNA section, we're the biology section. It encompassed not just DNA. At one point, back before DNA testing, there was other testing done on, it's called biological fluid.
And it's well known that handling and packaging wet evidence that may have biological evidence on it is how it should be done. I didn't say that at all.
Can you repeat the question?
Yes, it's well known in the process of collecting and preserving biological evidence how you should handle wet evidence.
Correct.
In fact, wet evidence that's believed to contain biological material is supposed to be air dried.
Yes, anything wet that might have blood or any kind of biological material on it should be dried down, because if it remains wet, that moisture, if you package it, that could get trapped in, and you could develop microorganisms, bacteria, mold, whatever, and all of those things degrade the DNA that might be there.
And in fact, it's recognized that there's a high probability that biological material will be destroyed by bacterial growth, which could potentially preclude the generation of DNA results if wet material isn't air dried.
That's correct.
So that brings us to the sweatshirts. Were you present at the time they were received by FDLE or soon thereafter?
The first time I saw the sweatshirt was when the package was brought to myself.
Had it been opened already?
Yes.
Who opened it?
Alvin Guzman, who is an analyst or forensic technologist in the trace evidence section, had already opened that package and done his examination, and then sealed that back in the package, and I got a transfer from him.
Trace evidence is another section of your lab?
Yes.
Just generally, that's where the evidence might be inspected for stuff. Whatever little stuff might be clinging to it, it would be collected and packaged and then perhaps submitted to someone else to analyze?
Yes. Trace evidence looks at like small fibers and stuff like that.
And when you got the evidence of the two shirts, were they repackaged and sealed in the same packaging that they were submitted to the lab?
Yes.
And what was that, please?
The outer container was a... May I refer to my notes?
Of course. Mm-hmm.
Okay. Both sweatshirts, the light gray one and the dark gray one, they were both packaged. The outer container was a sealed brown paper bag, and then inside that sealed brown paper bag was a red, what I call a biohazard bag. It's a red plastic bag that's usually used for putting biohazardous material in. And that biohazard bag was not sealed with tape necessarily, but it was knotted, sealed with a knot. And then inside that red bag was the sweatshirt, both sweatshirts, individually in separate packages, not in the same package.
It's common for you to receive evidence in brown paper bags, correct?
Yes, brown paper bags is ideal for packaging items because the paper bag lets the item breathe. So if there's any moisture in there, it'll dry out.
Unfortunately, in this situation, while the exterior packaging was brown paper, the interior packaging was plastic.
Yes, it was a red biohazard plastic bag.
And even though you didn't see it when Mr. Guzman opened it, you're saying that he tied it back up before he gave it to you?
Yes.
And then when you opened it to begin your inspection, you detected quite an odor of mold or ammonia,
Yes.
correct? It struck you that it smelled bad.
On one of the sweatshirts, the dark gray hooded sweatshirt. The light gray sweatshirt did not have the same odor.
And that meant to you that it maybe wasn't as wet?
Correct.
I take it your assumption, your professional assumption was this smell is associated with evidence that has been packaged wet and then stored for a period of time.
Correct. The items, both sweatshirts were still wet when I opened the package. The light gray non-hooded sweatshirt was damp, a little damp. It wasn't really that wet. The dark gray hooded sweatshirt was more damp. It was wet, and it had a very pungent odor. Ammonia, mold, I'm not exactly sure what the odor was. It was something along those lines. I noted in my notes.
Did you ever find your gym bag in the trunk after a couple of weeks? That kind of odor? You know what I'm talking about? Kind of a strong ammonia, mold, this dirty laundry that's been sitting around a while?
I've had sports equipment that have smelled like that before, yes.
What should have been done, if I hear what you're saying, is that the clothing should have been air dried before it was packaged, or at least packaged in paper bags.
That's correct. Ideally, yes, you'd like to air dry anything you collect, especially items of clothing that are wet. You'd like to air dry them before you package them. And on top of that, we encourage our agencies not to put things into plastic bags because those don't allow the evidence to breathe, and that'll trap in any kind of moisture.
And that would be a red flag to you as an analyst that there's something going on with this evidence that places the biological evidence at a risk of degradation.
Correct.
The things that you were talking about before, how the strands break up and such.
Yes, that's correct.
And I take it would be impossible for you to be able to quantify precisely how much degradation there was.
And that's correct. I mean, you can tell by the testing you do. Maybe you expect more results than you got, and you can say, oh, there was probably some degradation. However, I mean, that's a hypothetical situation. If the evidence were allowed to dry down and package properly, would I have gotten more results? Potentially, but I don't know for sure, and that's a hypothetical.
That's an unknown factor in this case.
Correct.
You don't know what you don't know.
Exactly.
So, did you dry the evidence at that point?
I left it out to dry as long as I could. However, I needed to transfer that item to a firearms examiner, Amy Seward. So I can't, when we transfer from one section to another section, I just can't take the sweatshirt and hand it to her, say, here you go. That needs to be put back into the original packaging for the transfer. So I did package that back up, and it was still wet when I packaged it back up. But that transfer from Alvin Guzman to myself to Amy Seward took place in a matter of hours, not days or weeks.
Had you taken your cuttings and finished your photography and done your swabbing before you passed it on to Ms. Seward?
Correct. I took everything I needed. I did all of my analysis because I knew that was going to be transferred on. And once another section gets a hold of that and they're testing it for latent fingerprints or firearms exams or whatever they're doing to it, that could potentially affect my ability to get results. There's always an order at which the evidence goes from section to section.
So the way the sequence went in the lab, Mr. Guzman started with the trace,
Correct.
and he wouldn't apply chemicals for that sort of thing.
No.
It's more of a visual inspection and then collection of fibers and such.
It's called sweeping, what he does, and I've never done it. I'm not trained in how to do it, but I have observed him doing it, and he's basically brushing the item to collect any kind of trace evidence, any kind of fibers or small strings or outside grass debris, whatever was on that sweatshirt, and packaging that into a small paper fold for any future examinations or testing.
And then it goes to you, and you don't do any chemical application to the fabric itself, or do you?
No. I perform that chemical test for the possible presence of blood. I rub a piece of filter paper and then test the filter paper. And if that's negative, I take a small cutting from the stain and I test directly on the fabric from that small cutting. I never actually drop any kind of chemicals onto the actual garment itself.
So you do a very careful inspection, no doubt, but it's to look for something that may be blood in your mind, or
Correct.
some biological evidence of some sort.
Correct.
And when you see it, you then use the filter paper, and then you may test the filter paper.
For the blood test, yes.
And that would give you the presumptive test that, hey, this is blood.
It gives me positive or negative result for the possible presence of blood.
If you get the negative, I think, as you've talked about on some of these items, you're satisfied that it's not blood, so you don't do any more testing typically.
It depends on the scenario and the item. But if I'm looking for blood stains on an item and something tests negative, then I'm not going to go on and do DNA testing on that stain.
And there are a number of items or number of spots or stains where when you got a negative with the rubbing, that was the end of your analysis as far as that spot was concerned.
Correct.
And then when you get a positive, you know it's positive for blood, but that then, in the sequence, you'll then do the DNA analysis on that spot. Do you go back and swab the spot, or do you test the filter paper?
No, I take a small cutting. I take a scalpel and I cut out a portion of that stain. As we were looking at the items down below before, you saw little squares missing from those stains. Those are the portions I cut out and put into a tube in order to do my testing.
I think I heard you say that the size of the cutting is somewhat proportional to how much surface area you think you need to get a good result.
Yes, in order to get a complete DNA profile, what I think I need.
So it starts with the visual, goes to the rubbing, and then to the cutting. Is that correct?
Correct.
And then when you do the cutting is when the DNA part comes in. That's what's processed for DNA.
Yes.
Can you describe briefly what that is? What do you do at the point that you've got these cuttings?
What do you mean? What
do you do with them?
I store them until I'm ready to start the first step, which would be the extraction process.
How does that work?
I'm applying heat and chemicals to the cutting in that tube in order to break open all the cells that are in there and expose the DNA molecule. And then I'm trying to wash away all that cellular debris and everything that's in there besides the DNA. I'm trying to get nice isolated DNA from that stain.
I know this sounds silly, but you can't see any of this, right? We're talking truly DNA.
No, I can't see DNA, but I can see a tube and I can see the chemicals in the tube. It's a small tube with a little bit of usually clear liquid. Various different chemicals are used.
And then when you go through your process, you at the other end get results. And then you're able to interpret those results as to whether you believe it's from a certain individual, maybe a number of individuals, and then whether or not you can make some sort of opinion as to who it's from.
Yes.
And that would be the process that you would have used for all of the samples for which you did DNA analysis?
Yes.
So, on the hooded sweatshirt, could we concentrate on your findings there, just for a moment?
Yes.
Stain. I suppose we should look at the shirt itself, if we could, for a moment. Would you be able to correlate the stain that's marked on the shirt and just say what you found as to each of those stains where you were able to make a finding?
Yes.
Well, let's do that then, if we could get some help. Let's start with the hooded sweatshirt. What's the best way for you to do this? Where do you want to be?
Not leaning against the door. Down there would be fine.
Can we sit it on the table again?
Front to back.
Front?
You got it?
You got it?
Yes, thank you.
You were talking about the hooded sweatshirt being one of the two items of clothing that was the wettest with the strongest odor.
Correct.
Can you tell me now, because I lost my note, where it was that the stain was that you identified as A?
Stain A is located here on the bottom, the front surface, on the front pockets.
And do you know whose blood that was?
Iris got a partial DNA profile, and that matched Trayvon Martin.
Did you exclude George Zimmerman from that?
Yes, it's a single source DNA profile, so when I match that to another individual...
I'm sorry, it's a single source DNA profile, so I match that.
When I match a single source DNA profile to an individual, that other individual is then excluded.
And you don't know, of course, how it came to be there, just that, in your opinion, that was a single source DNA sample or DNA from blood from Trayvon Martin.
When I test a blood stain, I have no way of knowing. There's nothing in my testing that tells me when or how a stain got on an item.
For example, it could have transferred from blood on some other part of the shirt, depending on how the shirt may have been positioned at different points in time.
Potentially, yes.
Let me grab that chart. Maybe that will help me better to be more precise. Was there any other blood? Stain D. Where is stain D?
There was A, B, and C on this item. B and C are on the back.
All right, that's my confusion. Is this what's known as the gray sweatshirt?
This is the hooded.
No, I'm really disoriented here. What stains are on the front, and who are they from?
A, partial profile matched Trayvon Martin. B and C are on the back.
And whose are B and C, if
B
you know?
tested negative for the possible presence of blood. Performed no DNA testing on that. C, I obtained positive for the possible presence of blood. No DNA results were obtained from that.
So the only stain on the outside, or the gray hooded sweatshirt that you could match to a DNA profile is A on the front?
Correct.
And were there any other stains where there might have been a partial profile that you couldn't include or exclude Trayvon Martin or George Zimmerman?
No, the only stains I got tested for DNA were A and C. And C, I got no DNA results. I did swab as I explained earlier, the sleeves and cuffs of this item as well.
Tell us again what your I know what your process was. You just swabbed it basically from the elbow down to the cuff.
Yes.
Not having seen any blood, correct?
Correct. I first used the filter paper to rub it and test that for the possible presence of blood, and that was negative on both sleeves. So I performed the swabbing, DNA testing, and there was no DNA foreign to Trayvon Martin on either of those.
Is that looking, though, if it's not blood, and you're pretty sure it's not blood, are you looking for skin cells or some other source of DNA, perhaps?
Yes, any foreign DNA that might be on these sleeves, which could be touch or skin cells, it could potentially be saliva, so anything foreign to Trayvon Martin, I was looking for potentially.
Frankly, then, you just didn't find much of anything on this gray hooded sweatshirt. It boils right down to it.
Just that partial profile on stain A.
That was the shirt that stunk to high heaven.
It didn't smell good, no.
Let's go to the next item if you could. Give us a moment. We'll switch out. You've described as the gray sweatshirt?
Yes.
Let me get the exhibit number, if I might, so that...
156. 156.
Oh, okay. 156. And the gray hooded sweatshirt, 155. All right, 156 has been sort of represented as the underneath shirt, correct?
Then there was no representation as far as my end. They were just two sweatshirts.
But this one was quite a bit drier.
Yes.
It didn't smell as bad. It wasn't as obviously wet.
It was slightly damp.
So, same question. Sorry to be so imprecise. Can you tell me where the blood is and whose it is on the front of this gray sweatshirt?
Can I get my report
Of course.
so I don't get them confused?
Of course.
All right, there were stains A through E that I tested for blood.
Let's talk with... Go ahead.
Stain C tested negative for blood. I did not perform DNA testing on that one. I performed DNA testing on stain A, which was
All right.
positive for blood.
Let's talk about that a second. Is that reddish-brown stain on the lower right-hand side of the shirt?
Yes.
Pretty visible, I take it?
Yes.
I mean, it was obvious to you that that was a stain that would be of interest to you.
Yes.
And there's a small hole in it. Is that the cutting that you took?
Yes.
Basically, that's all you thought you would need to get a positive reading on that stain.
To get a complete DNA profile, hopefully. Yes.
Again, you don't know how it came to be there, but you know it was there when you saw it.
No, I don't know when and how.
And whose blood is that?
That I obtained a complete DNA profile which matched George Zimmerman.
Is that a single source?
Yes.
Only George Zimmerman.
Yes. There was the possibility of additional contributor to that profile in stain A, and what that means is there was a very, very small amount of potential DNA that wasn't even high enough for me to be able to interpret. So I did report out that there was the potential of additional contributor. However, that's nothing that I could ever make a comparison on. It's just a couple little blips, we call them, that I had to report out.
Okay. Continue, if you would.
Stain B, I also tested, and that was a complete DNA profile as well, and that matched Trayvon Martin.
Where's B located?
B is also on the bottom, small stain.
More in the center.
Yes.
When I see that, I don't see any reddish substance at all in what you've circled there.
Yes,
Why not?
because I cut it all out.
So you, in your cutting, you took all of what you could see, maybe blood?
Yes, if there's a very, very small blood stain and it tests positive for blood, if I feel like I need to consume that stain and cut the whole stain out for maybe to be able to potentially get a profile to do something with it, then I do that. And other examples where there's a good sized blood stain and I don't have to take the whole stain, that's what I do.
So A is George Zimmerman, B is Trayvon Martin.
Yes.
Is that a mixed sample?
No, single source.
And let's just go alphabetically. C, you said, was negative.
Correct.
Was not blood.
Correct.
And that brings us to D.
D was positive for blood as well. That was a mixture of at least two individuals. I was not able to resolve that into any individual major or minor DNA profiles. And both Trayvon Martin and George Zimmerman were both included in that mixture.
Can you look at the area that you're calling D, and can you still see some staining that you think may be blood?
Yes. There's still some reddish area, reddish-brown area there.
Like you did with A, did you just get enough of that stain to do your DNA testing?
Correct.
And there are 2 spots, it looks like. No, I'm sorry, 3 or 4, aren't there? Each of those defects from cuttings that you did?
It appears so. Stain D was a very light, spread out stain, so I took what looks like 4 cuttings and combined those into 1 tube to try to give me the best, the potential best DNA profile I could get.
All right, follow with me a second. So it's possible then, I take it, what you cut here was Trayvon Martin or George Zimmerman?
Yes. Obviously, in hindsight, having a mixture of DNA, maybe I could have taken, 1 cutting from here and tested that separately from the cutting from here. But that's in hindsight. I don't know at the time. It looked like one big light-colored stain, so I took several cuttings and combined it in 1 tube to try to get a complete DNA profile. Obviously, knowing what I have in the end, going back to it, maybe I could have taken those cuttings separately and gotten single-source individual profiles on the separate items. It's potential, yes.
Just, for example, you might have been able to identify one or the other as from that particular cutting.
Correct.
Or from any of the others.
Correct.
But by doing them all at once, you got a full view of the DNA in those cuttings.
Correct.
And then you were able to determine that it was a mixed sample?
Yes.
And it was both Trayvon Martin and George Zimmerman?
They were both included in the mixture.
That's D. Anything else on the front?
Stain E.
Yes.
Also
That's
positive for blood.
Under the left armpit?
And that matched, complete DNA profile that matched Trayvon Martin.
I know that you also talked about the sleeves.
Yes.
You didn't see anything on the sleeves that you thought was obviously blood.
Correct.
Nothing that you would use filter on because you saw a spot.
Correct.
So tell me again what your process was?
I took a piece of filter paper and rubbed the entire lower part of the sleeve and cuff, and that tested negative for blood.
And that's separately?
Yes, both sleeves separately.
Or negative?
Negative.
So what's next? What can you do next for non-blood DNA?
Just take a cotton tip swab and swab it for any foreign DNA. That would be touch skin cells or potential saliva.
So let's talk about the left and the right. Just start with the left. What were your findings
left
there?
Sleeve or cuff. I did get foreign DNA to Trayvon Martin. However, they were limited foreign results that I could not match to anyone, and I was not able to determine if George Zimmerman was a foreign contributor to that sample.
Is that, as you said before, you can't say he was or can't say he wasn't the contributor of that DNA?
Correct. It's an in-between state.
So it could have been, you just can't say it was.
I wasn't able to exclude him.
And how about on the right side?
I was not able to get anything foreign to Trayvon Martin on the right sleeve.
Does that mean you were able to find DNA that was attributable to Trayvon Martin?
Yes.
But you were looking really for DNA that wasn't.
Correct.
Anything else on the front?
No.
And let's just take a look at the back. I don't remember there being stains on the back, are there?
No.
Just display this for a moment, and then we'll take it back. Mr. Gorgone, looking at the back here, there's no cuttings, no markings. Can't get good help these days. Nothing here that
There were
you
no
analyzed.
No cuttings that I took from the back.
Great. Okay. And I do want to talk with you, though, briefly about George Zimmerman's outside jacket, the orangish-red jacket.
Yes.
Without going through all of the samples that were George Zimmerman, were there any spots on that outside shirt that were or could have been Trayvon Martin's?
On the orange jacket? Yes.
And let's do this one more time. You can point that out. The witness may step down.
Yes.
Thank you, Judge. Let's just have you focus on the stain or stains that were or could have been Trayvon Martin's.
The ones I matched or included Trayvon Martin on?
That you matched or couldn't exclude.
Okay. Stain E, which I don't believe is on the front. Stain N, which is on the front, located right here on the front right shoulder. That was a mixture. That was the mixture that I was able to resolve a major as well as a minor contributor. The major contributor matched George Zimmerman. The minor contributor matched Trayvon Martin.
When you say major and minor, you're talking about more from one, more blood from one than the other?
Well, more DNA from one than the other. Major would be the more DNA, minor would be the lesser DNA.
Can you still see some of the staining from N that you talked about on the jacket?
Yes.
The cutting that you took in the same fashion you'd done before was a visible stain that was positive for blood with the filter paper rubbing,
Yes.
and is representative basically of what you thought you would need to get a reliable DNA result.
Yes.
You didn't test any of the other specific spots.
The whole stain was rubbed with the filter paper for the blood test.
Right.
But
Just-
as far as the DNA testing,
Mhm.
these were the two cuttings that I took and put in a tube. There's one clearly visible, and there's actually one on the interface, so to speak, between the two colors.
So sort of like stain D, I
Yes.
think. looking back, you may have chosen to make separate cuttings within the larger stain, but in this case, the DNA results were a combination of both of the cuttings.
Correct. It's possible that one cutting was could have been an individual profile in this one, but it's possible they both would have been mixtures, regardless of whether I tested them separately or together.
But there's no question in your mind, as an FDLE DNA analyst, that was a mixed stain that contained both the DNA of George Zimmerman and Trayvon Martin.
Both profiles matched the two individuals.
Anything else on the jacket that relates to that it was Trayvon Martin's blood or that he was not excluded?
Stain U, which is right here, if you can see on the bottom of this cuff. That's stain V here, but on the bottom is stain U. That was also a mixture.
The right cuff, am I correct?
That is the right cuff. That was also a mixture of DNA that Trayvon Martin was included as a possible contributor. And I did not make any determinations on George Zimmerman to that specific stain.
Same sort of technique, you rubbed it and
Yes.
then made cuttings?
Yes. Stain V, which is right next to stain U, that one was too limited for me to be able to make a match. However, I was not able to exclude either of the two individuals.
V could, from the limited information you had, could have been Trayvon Martin. You couldn't say it wasn't, but you can't say it was.
Correct.
V is represented by this larger circle with the three separate cuttings?
Yes. That's it for the front.
Okay. Maybe just flip the exhibit. Now we are looking at the back of the jacket in Exhibit 151.
There were two on the back. I was not able to make a determination if Trayvon Martin was present.
Excuse me a moment, I'm just going to pass. Thank you. All right. Thank you, go ahead.
Stain E, which is right up here on the back of the right shoulder, the right-hand shoulder, with the two cuttings. That one was a mixture where I was able to resolve a major. I matched that major, complete major, to George Zimmerman, and I was not able to make a determination or exclude Trayvon Martin. And then the last one was stain double B, which is also here in between the two colors. That one was also a mixture. That George Zimmerman was included in that mixture, and I was not able to make a determination on Trayvon Martin or exclude him.
Okay. Is that it?
That was it.
We'll put this back. You may take your seat, please. Thanks. Your Honor, if I may have just a moment, I'm nearing completion here.
Yes, indeed.
Looking for. Thank you, Mr. Gorgone.
Thank you. Redirect.
Yes, Your Honor.
what's been identified as the defendant's jacket, the red jacket, red and black jacket?
Yes.
The one you just talked about? You mentioned exhibit stain U, I think it was,
Yes.
and you mentioned that you said the right cuff, right? Did I get that right?
It was on the right cuff, the bottom of the right
Okay.
cuff.
And you said Trayvon Martin could be included as a possible contributor, correct?
Correct.
Okay. May I approach the witness, Your Honor?
Yes, you may.
Okay. Let's assume I'm wearing that red jacket, correct?
Okay.
And so you're saying on the right side, right here, some of Trayvon Martin's possible DNA is there, right?
Potentially, yes.
Potentially. I'm just saying he's included as a possible, correct?
Yes.
Okay. So if you're Trayvon Martin, may I approach the witness, Your Honor?
Yes,
And I promise I won't hit you.
I'm sorry, there's an objection.
To a hypothetical with the witness beyond the scope of cross that is asking this witness to speculate about an area for which he said he has no expertise. He's a DNA analyst, not a...
Okay, that's a speaking objection.
Yes, ma'am.
So if you want to approach, you may do so.
Right jacket. You're saying on the defendant's jacket, or George Zimmerman's right jacket, correct?
The right arm.
I'm sorry,
Yes.
right arm on the cuff, you said there could be Trayvon Martin's DNA, correct?
Correct.
Okay. And so that had to have, if it was Trayvon Martin's potentially, it had to have come into contact with Trayvon Martin, correct?
Objection. This witness is not qualified to answer that question because of all sorts of
Okay,
other...
no speaking objections, but he could testify if he knows.
In other words, that had to be deposited if it was Trayvon Martin, so if he's included as a possible, it had to get there somehow, correct?
It had to have come into contact with some kind of bodily fluid
Of
from
Trayvon Martin.
Trayvon Martin or skin cells from Trayvon Martin.
Right? Correct?
Correct.
And I think Mr. West asked you twice, You don't know what you don't know, right? Remember
I don't
him
know
telling
what
you
I
that?
don't know. Yes, correct.
Okay. In other words, there could be a rat right under this table, but you don't know, right? That would be pure speculation,
Correct.
right? Unless you came down here and looked and saw a dead rat or did see a dead rat, right?
Correct.
But what you can unequivocally say as to the defendant's jacket, that in his right cuff, Zimmerman's jacket, there could be, potentially, there's DNA there, and it could be Trayvon Martin's.
He was included.
He was
in the-
included as a possible. I apologize.
He was included in the mixture I obtained from the bottom of that right cuff.
Now, how it got there, you don't know because you weren't out there.
Correct.
Could be from all sorts of possibilities.
Correct.
But definitely, it made contact with potentially Trayvon Martin's DNA.
Objection. That's a confusing question, if nothing else.
Okay, do you understand the question?
Yes.
Then you may answer.
Please.
He is included as a possible contributor, and if his DNA is, in fact, part of that mixture, it would have to have come in contact with some kind of bodily fluid or skin cells for me to have been able to get that result from that swab or a cutting of that cuff.
And you also still focusing on the red and black jacket. You also said that stain U, I think? Or did I cover? I'm sorry, stain R? Did I get that right, or have I got?
No.
I'm sorry, stain U was the right cuff. If I got that right?
Stain U was the one we were just talking
Right,
about.
but it's the right cuff, meaning right here, right cuff. Correct?
Right cuff on the right hand.
I'm sorry, on the right hand. I apologize. And I think you were also asked about the victim's, Trayvon Martin's hooded sweatshirt, the one that, assuming he wore that over, the regular sweatshirt.
The dark gray hooded
Yes,
sweatshirt.
with the hoodie,
Yes.
correct? Now, did I understand you correctly that you examined that and you did not find any of George Zimmerman's DNA on that?
There was nothing that matched, or he was not included in anything from that sweatshirt.
So in other words, you tested, I think you said 5 different or 3 areas, A, B, and C, and then you also tested his right sleeve and his left sleeve, the hooded jacket's right sleeve and left sleeves, cut right from here to here, and you didn't find any of George Zimmerman's DNA on there, correct?
Objection. Asked and answered. Pardon me? Yes.
Right, overruled.
You understand my question, sir? I'm gonna
Yes.
repeat it. Okay. You did not find on Trayvon Martin's hooded, you tested from here to here, and you didn't find any of the defendant's DNA on there, correct? George
There was
Zimmerman.
nothing.
Okay, now it's been asked and answered, it's the same.
And on the other parts, A, B, and C, you didn't find the defendant's or George Zimmerman's DNA, right? On the other parts of the hoodie.
I didn't test B, but A and C, there was nothing that matched him. No.
And B was negative as to blood.
Correct.
Thank you. No further questions.
Mr. Gorgone, thank you. Addressing your attention to the right cuff area of Mr. Zimmerman's red jacket. Stage 151.
Yes.
Your testimony is that there's DNA there that you were able to identify.
Yes,
You don't know how it came to be there, I take it.
correct.
You don't know if it was connected with the gunshot itself, or whether Mr. Martin may have had an abrasion on his knuckle.
Objection, now it's a speculation as to,
Oh, let me hear the rest of the question. Go ahead and continue.
That Mr. Martin may have an abrasion on his knuckle that was bleeding, that came in contact with Mr. Zimmerman's cuff. You just wouldn't and don't want to speculate how it came to be there.
All I know about that sample is it tested positive for the possible presence of blood, and I obtained a mixture in which Trayvon Martin was included in.
Thank you.
Any redirect?
no, Your Honor.
May Mr. Gorgone be excused?
Yes, Your Honor.
Okay, thank you very much, sir. You are excused. Yes. Ladies and gentlemen, we're going to go ahead and recess for your purposes for this evening. I would like to start Friday morning at 8:30. When you're the overnight and Thursday recess, you're not to read or listen to any radio, television, or newspaper reports about the case. You're not to discuss the case amongst yourselves nor with anybody else. You're not to use any type of an electronic device to go on the internet to do independent research about the case, people, places, things, or terminology. And you're not to read or create any emails, text messages, Twitters, tweets, blogs, or social networking pages about the case. Do I have your assurances that you will abide by these instructions? Okay, enjoy your evening. Have a good 4th of July. We'll see you Friday morning at 8:30. Please put your notepads face down.